Assalamu alaikum, dear listeners. From the series of lectures on human digestive system, in our last lecture, we have gained considerable knowledge on gastrointestinal tract and its accessory organs, their functions, histology, regulation, and their secretions such as enzymes and hormones. In our current lecture, we will talk about digestion, absorption, and transport of carbohydrates. So let's start our lecture. We know that digestion is a process involving the hydrolysis of large and complex organic molecules of food that we eat into smaller and preferably water-soluble molecules, which can be easily absorbed by the gastrointestinal tract for utilization by the organism. So, digestion of macromolecules also promote the absorption of fat-soluble vitamins and some certain minerals. So, if we talk about carbohydrates here, as we know that there are three types of major macromolecules: carbohydrates, lipids, and proteins. So, if we talk about carbohydrates, the major source of carbohydrate which is found in plants. The principal dietary carbohydrates, they are polysaccharides that contain starch and glycogen. Starch in plants and is a stored form of carbohydrate that is present in plant, and glycogen is a form of stored carbohydrates present in animals. Then are disaccharides that contain the examples are lactose, sucrose, and the third type monosaccharides. The examples are glucose, fructose, etc. The glucose concentration in the body, they are maintained within limits by various metabolic processes. The digestion of carbohydrates occur in the mouth and intestine. The hydrolysis of glycosidic bonds is carried out by a group of enzymes which are called glycosidases. It's important to understand what is a chemical digestion. So what is a chemical digestion? When it comes to digestion, chewing is only half the process. As food travels from mouth into digestive system, it is broken down by different digestive enzymes and turned into smaller nutrients or smaller particles that our body can easily absorb. Now this broken down is known as chemical digestion. Without this chemical digestion process, body wouldn't be able to absorb nutrients from the foods that we eat. So it's important to learn more about chemical digestion, including how it's different from mechanical digestion. So the question arises, how is chemical digestion different from mechanical digestion? Chemical and mechanical both digestions are the two methods that our body uses to break down foods into smaller particles. Mechanical digestion actually involves physical movements to make foods much smaller, while chemical digestion uses enzymes and other chemicals like HCl to break down food particles. So mechanical digestion, it begins in mouth with chewing, then moves to the churning in the stomach and segmentation in the small intestine. We have already seen the phenomena of chewing, churning and segmentation in the last lecture of digestive system. Peristalsis, it's a movement which is also part of the mechanical digestion. This refers to the involuntarily contractions and relaxations of the muscle of the esophagus, stomach, and intestine to break down food and move it through the digestive system. So we can say that mechanical digestion is carried by chewing, churning, segmentation, and peristaltic movements of gastrointestinal tract organs. Then comes chemical digestion. Chemical digestion involves the secretion of enzymes throughout the digestive tract. These enzymes break the chemical bonds that hold food particles together. For example, glycosidic bond in carbohydrates, and this allows food to be broken down into small and digestible parts. Now the next question is how they work together. That is both of the types of digestion, how they work together. Once food particles reach the small intestine, the intestine continue to move. This helps keep food particles moving and expose more of them to digestive enzymes. These movements also help to move the digestive food toward the large intestine for eventual secretion. What is the purpose of chemical digestion? Digestion involves taking large portions of food and breaking them down into micronutrients which are small enough to be absorbed by cells. Chewing and peristalsis both help with this process, but they do not make particles small enough. That's why we need chemical digestion, which comes here. Chemical digestion breaks down different nutrients such as proteins, carbohydrates, and fats into even smaller particles. So we can say that mechanical digestion converts macronutrients towards micronutrients, and chemical digestion converts these micronutrients towards their units, such as fats will be broken down into fatty acids and monoglycerides. Monoglycerides, they are units of fats. And similarly, nucleic acids, they will be broken down into nucleotides. Polysaccharides or carbohydrate sugars, they would be broken down into monosaccharides, and proteins will be broken down into amino acids. Without chemical digestion, body wouldn't be able to absorb nutrients, leading to vitamin deficiencies and malnutrition. Because chemical digestion produces micronutrients, but micronutrients cannot be absorbed directly from the walls of the small intestine and large intestine. So, where does chemical digestion begin? Chemical digestion begins in the mouth. As the food is chewed, the salivary glands release saliva into the mouth. The saliva contains digestive enzymes such as salivary amylase that start the process of chemical digestion. Digestive enzymes which are found in mouth, they include lingual lipase and salivary amylase. Lingual lipase, this enzyme breaks down triglycerides, which is a kind of fat, and salivary amylase, this enzyme breaks down polysaccharides, which is a complex sugar and is a carbohydrate. What path does chemical digestion follow? Chemical digestion doesn't just stop with enzymes in the mouth, but rather it follows the gastrointestinal organs, such as first of all stomach, then small intestine, large intestine. There are also sensory organs that take part in the process of chemical digestion, such as liver, pancreas, gallbladder. So, chemical digestion is a vital part of the digestive process, without which our body wouldn't be able to get proper energy for daily work and activities. Digestion of carbohydrates. The principal sites of carbohydrate digestion are mouth and small intestine. We know that monosaccharides need no digestion prior to absorption. Why? Because they are simplest molecules and the key units of the carbohydrates. Whereas disaccharides and polysaccharides must be hydrolyzed to simple sugars before their absorption. The hydrolysis of glycosidic bond is carried out by a group of enzymes which are called glycosidases. There are monosaccharides. As we know that carbohydrates has three classes: monosaccharides, the simplest one, disaccharides, and polysaccharides. Here are some examples of monosaccharides which include glucose, galactose, and fructose. They contain a single molecule. Then comes disaccharide. Here are some examples of disaccharides which contain sucrose, lactose, and maltose. Sucrose is made up of glucose and fructose, while lactose is made up of two monosaccharides, glucose and galactose, and maltose is made up of two monosaccharides, glucose and glucose. So when disaccharides will be broken down, such as if sugar will be broken down by the action of amylase of enzyme sucrase, it will be broken down into glucose and fructose. Similarly, lactose will be broken down by the action of enzyme lactase into glucose and galactose. And maltose will be broken down by the action of enzyme maltase into glucose and glucose. Here is a process known as condensation. This process is used to convert monosaccharides into disaccharides. So the chemical reaction takes place by the involvement of hydroxyl group which are present on glucose and fructose. Here we can see that a hydroxyl group from fructose and a hydrogen atom from glucose, both of them combine and condense to form a water molecule. This water molecule is released and ultimately these monosaccharides, glucose and fructose, they are condensed towards a disaccharide which is known as sucrose. Next chemical reaction is a hydrolysis. The process is known as by the addition of water. This chemical reaction is opposite to the condensation reaction. In hydrolysis, we can observe that a water molecule is added to a disaccharide where one monosaccharide where hydroxyl group is added to a glucose molecule and hydrogen is added to another glucose molecule, and ultimately a disaccharide is converted into two glucose molecules. Condensation and hydrolysis, both of these reactions are used in the process of absorption and digestion. Amylose. Amylose is a form of starch. Glucose molecules are stored in the form of starch in plants, while glycogen, it's a form of storage form of glucose molecule. Glucose molecules are stored in the form of glycogen in animals. We can observe from these diagrams that starch is relatively a simple molecule as compared to the glycogen. Glycogen is a complex molecule because it produces more energy as compared to starch, as animals need more energy for their daily and routine work as compared to plants. This diagram shows an overall description of chemical digestion and absorption of dietary carbohydrates. If we start from different classes of dietary carbohydrates such as monosaccharides, disaccharides and polysaccharides, we can here observe that monosaccharides needs no chemical digestion and would be directly absorbed in the bloodstream, while disaccharides being converted to being containing two molecules of monosaccharides would be converted to first of all monosaccharides by the action of chemical digestion, that is enzymes, and would be converted to monosaccharides such as lactose, sucrose or maltose. These monosaccharides would be then absorbed in the bloodstream. Then if we talk about polysaccharides would pass from mouth and by the action of salivary amylase they would be converted to dextrins. Dextrins pass through stomach and remain untreated as no digestion occurs in stomach, then these dextrin molecules will enter the small intestine and would be acted upon by pancreatic alpha amylase and would be converted to disaccharides. These disaccharide molecules then by the action of intestinal enzymes include maltase for example would be converted to monosaccharides, and then these monosaccharides would be absorbed in the bloodstream. Digestion in mouth. The salivary glands in the oral cavity secrete saliva that coats the food particles. Saliva contains the enzyme salivary amylase. This enzyme breaks the bonds between the monomeric sugar units of disaccharides, oligosaccharides and starches. The salivary amylase breaks down amylose and amylopectin into smaller chains of glucose called dextrins and maltose. As we have seen in the last slide, the increased concentration of maltose in the mouth that results from the mechanical and chemical both types of digestion, the breaks down of starches in whole grains is what enhances their sweetness also. Only about 5% of starches are broken down in the mouth. So this is a good thing, as more glucose in the mouth, would lead towards more tooth decay. So we can say that saliva contains carbohydrate splitting enzyme, salivary amylase, also known as ptyalin. Action of by the which initiates the hydrolysis of a starch. By the action of salivary amylase, that is ptyalin, it is the alpha amylase which requires chloride ions for activation. Chloride ions are required for activation of alpha amylase, and optimum pH, that is 6.7, almost 6.6 to 6.8 pH is required for the action of alpha amylase. And salivary amylase so hydrolyzes alpha 1,4 glycosidic linkage of polysaccharides and producing smaller molecules like maltose, glucose, trisaccharides, and maltotriose, etc. It is important to note here that alpha amylase does not digest alpha 1,6 bonds in the mouth. Then comes digestion in stomach. Carbohydrate digestion. When carbohydrates reach the stomach, no further chemical breakdown occurs because the amylase enzyme does not function in the acidic conditions of the stomach. As we have seen in our last slide that the pH, the optimum pH for the action of salivary amylase is around about 6.6 or 6.7. So in stomach, we know that the pH is around about 2 to 3, and so alpha amylase become inactivate in such low pH, in such acidic conditions. But mechanical breakdown of carbohydrate is going on in stomach. The strong peristaltic contractions, for example, of the stomach, they mix the carbohydrates into the more uniform mixture of chyme. So we can say that salivary amylase action stops in stomach when pH falls to round about 2 to 3, and no carbohydrate splitting enzyme are present in gastric juice. Similarly, some dietary sucrose, however, some dietary sucrose may be hydrolyzed to their equimolar amounts, that is very little amounts of glucose and fructose by the action of HCl. HCl also performs small amount of chemical digestion. Then comes digestion in intestine. The chyme is gradually expelled into the upper part of the small intestine. Upon entry of the chyme into the small intestine, the pancreas releases pancreatic juice through a duct. This pancreatic juice contains the enzyme pancreatic amylase, which starts again the breakdown of dextrins into shorter and shorter carbohydrate chains. We can say that this is the same amylase that that was already present in saliva, but here there in saliva the name was given as salivary amylase, but in intestine the name is given as pancreatic amylase. Additionally, enzymes are secreted by the intestinal cells. So we can say that there are two phases of intestinal digestion. Number one, the digestion due to pancreatic enzymes, and number two, the digestion due to intestinal enzymes. The pancreatic enzymes include alpha amylase, and intestinal enzymes include sucrase, maltase, lactase, and isomaltase. All of these enzymes, they are present in the villi cells of the small intestine. These enzymes, known collectively as disaccharidases, they are known as sucrase, maltase, and lactase. We can observe that, or we can say that sucrase breaks down sucrose into glucose and fructose. Maltase break maltase breaks the bond between the two glucose units of maltose. Similarly, lactase breaks the bond between galactose and glucose. So, one carbohydrates are chemically broken down into single sugar units. They are then transported into the inside of intestinal cells. So, if we talk about digestion due to in the duodenum due to pancreatic amylase, digestion of food bolus in duodenum mixes with pancreatic juice. Pancreatic juice contains pancreatic amylase, similar to salivary amylase. Action of pancreatic amylase is it works in optimum pH around about 7.1 and requires again chloride ions, as does the salivary amylase for its activation. It specifically hydrolyzes alpha 1,4 glycosidic bond and not hydrolyze alpha 1,6 bonds. It produces disaccharides, maltose, isomaltose, and oligosaccharides. Then comes digestion in jejunum. The final digestion of di and oligosaccharides to monosaccharides primarily occur at the mucosal lining of the upper jejunum, carried out by oligosaccharides. For example, glucoamylase here acting on amylose, and disaccharides, for example, maltase, sucrase, and lactase, they act upon disaccharides. Then comes the function of pancreatic amylase is to degrade dextrins further into a mixture of maltose, isomaltose, and alpha limit dextrin. While the alpha limit dextrins, they are smaller oligosaccharides and contain 3 to 5 glucose units. Then comes the second intestinal juice. Intestinal enzyme amylase. It hydrolyzes terminal alpha 1,4 glycosidic bond in polysaccharides and oligosaccharides and liberate free glucose. So apart from intestinal amylase, there are some other enzymes that take part in the chemical digestion, such as maltase. It hydrolyzes a alpha 1,4 glycosidic bond between glucose units in maltose, and its pH range is 5.8 to 6.2 roundabout. Then isomaltase. It catalyzes a alpha 1,6 glycosidic bond. It's important that isomaltase catalyzes alpha 1,6 glycosidic bond, which is not catalyzed by salivary or pancreatic amylase. Branching points. So alpha 1,6 glycosidic bond makes branching points. So branching points are degraded and broken down by the action of isomaltase and produce maltose and glucose. Then comes sucrase. Sucrase, it hydrolyzes sucrose to glucose and fructose, and its pH range is 5 to 7 around. Then comes lactase. Lactase is a beta galactosidase enzyme. Its pH range is 5.4 to 6. It is known as beta galactosidase because it breaks down galactose from the beta position. Then comes dextrinase. Dextrinase enzymes act upon alpha limit dextrins and convert them into glucose and maltose. Now we will study about absorption of carbohydrates. That is going of the monosaccharides towards the bloodstream. How they absorb, where absorption takes place. The cells in the small intestine have membranes that contain many transport proteins in order to get the monosaccharides and other nutrients into the blood, where they can be distributed to the rest of the body. So the first organ that receive glucose, fructose, and galactose is the liver. The liver takes them up and converts galactose to glucose, breaks fructose into even smaller carbon containing units, and either stores glucose as glycogen or exports it back to the blood. So we can say that absorption starts from small intestine. Small intestine sends all monosaccharides towards their required or target organs, such as liver and muscles. First of all, liver receives these monosaccharides. Now the question is how much glucose the liver exports to the blood. It is under the hormonal control and it depends upon our activities and our physical exercise. Two mechanisms are responsible for the absorption of monosaccharides: active transport and facilitative transport. Active transport means against movement of monosaccharides against a concentration gradient, that is from a low glucose concentration to a higher concentration, while facilitative transport means with concentration gradient, that is from movement of monosaccharides from a higher concentration to a lower concentration. This process, the facilitative transport, does not need energy as compared to the active transport, which needs energy for in the process of transportation. It is observed that glucose accounts for 80% of the total monosaccharides. The absorption occurs mostly in the duodenum and upper jejunum of small intestine. Only monosaccharides are absorbed by the intestinal wall, and absorption rate is maximum for galactose, moderate for glucose, and minimum for fructose. We will see absorption rate in the next slides. Absorption rate. Different sugars possess different mechanisms for their absorption. According to Corey, a scientist who studied the rate of absorption of different sugars from small intestine, he performed his experiments in rat. According to him, if we take glucose absorption as 100, then the comparative absorption for other sugars would be as follows. Galactose as 110, it means galactose is absorbed more rapidly as compared to glucose. Glucose is 100. Fructose 43. Fructose is absorbed in much less quantity. Mannose 19, xylose 15, and arabinose 9. Galactose is absorbed more rapidly than glucose, and pentoses, they are absorbed relatively slowly. Here comes mechanism of absorption. The slide shows how monosaccharides are absorbed from the walls of small intestine into the blood. Monosaccharides, the end product, which are end products of carbohydrate digestion, they enter the capillaries of the intestinal villi. We can say here that the small intestinal wall, the detailed elaborated diagram of a small portion of the small intestine, we can see here that the monosaccharides, they are absorbed and travel to the liver via the portal vein. So they are absorbed through the walls of the small intestine and are entering to the portal, to the arteries and veins, and then from these small veins they are collectively enters to the portal vein, from where they are transported to the liver. In the liver, galactose and fructose, they are converted to the glucose then. The sodium independent transporter named as GLUT2 that facilitates transport of sugars out of the mucosal cell of the intestinal wall, thereby entering the portal circulation and being transported to the liver. When the process is shown here in detail, when a monosaccharide molecule, such as sugar or another monosaccharide molecule, which comes in contact to the intestinal wall, it is carried by the transporter, the appropriate transporter, which then this transporter then binds this monosaccharide molecule and changes its direction and transfer this molecule to the bloodstream. After leaving the monosaccharide molecule, this transporter automatically turns back to its original position and is ready to carry another molecule, another monosaccharide in the similar way. Then comes the active transport. The transport of glucose and galactose across the brush border membrane. Here is the brush border brush border membrane of the mucosal cells. It is occurred, the transport of glucose and galactose occurred by an active transport. Active transport is an energy requiring process that requires a specific transport protein and the presence of sodium ion. As far as glucose receptors are concerned, there are specific glucose receptors to carry specific monosaccharides such as glucose, galactose and sodium. Both of these, they are carried by a sodium dependent glucose transporter carrier SGLT1, while fructose and mannose, they are transported by a carrier known as GLUT5, while glucose on the other side is carried towards the bloodstream with the help of another carrier GLUT2. All of these are glucose transporters through the intestinal walls from lumen towards the bloodstream. So if we look into the process in detail, what happens at the intestinal lumen and brush border villi, we found that glucose and sodium, glucose and sodium here, they share the same transport system, that is sym porter, that is referred to as sodium dependent glucose transporter SGLT1. The concentration of sodium is at first higher in the intestinal lumen as compared to the mucosal cell. So, sodium moves into the cells along its concentration gradient, and simultaneously glucose is transported into the intestinal cells. It means that sodium carry glucose along with it. While here sodium carries along the concentration gradient, that is from higher concentration towards lower concentration, but glucose travels from lower concentration towards higher concentration, that is against concentration gradient. So both of them, glucose and sodium, they are mediated by the same carrier system. Now sodium diffuses into the cell and it drags glucose along with it. The intestinal sodium gradient is the immediate energy source for glucose transport here. This energy is indirectly supplied by ATP. Since the re-entry of sodium, that is against the concentration gradient, into the intestinal lumen from here towards intestinal lumen is an energy requiring active process. Here, the enzyme sodium potassium ATPase is involved in the transport of sodium in exchange of potassium ions against the concentration gradient in order to balance the ionic concentration of the intestinal lumen across the intestinal epithelium. So at the intestinal lumen, absorption by SGLUT, and while at the other end towards the bloodstream, the absorption is carried by GLUT2 in the same way. In this diagram, we can look upon the same mechanism in much more detailed way. From the intestinal lumen, two atoms of sodium, they are carried in the intestinal epithelium wall from higher concentration towards lower concentration along concentration gradient and carry glucose and drags glucose along with it. Now the glucose enters the intestinal epithelium with the help of a transporter SGLPT, and then with the help of another transporter GLUT2, this glucose molecule is carried towards the bloodstream, that is along the concentration gradient from higher concentration towards lower concentration, as we have now low concentration of glucose in the blood. While here sodium is expelled by sodium potassium ATPase pump, where energy is provided by ATP molecule, and at this site two potassium atoms, they are forced to enter in the intestinal epithelium in order to regulate the concentration of positive ions. The potassium atoms are not shown here, but they are shown here in this diagram. Then comes facilitative transport. Fructose and mannose, they are transported across the brush border by a sodium dependent facilitative diffusion process. This facilitative diffusion process does not need any energy because here fructose and mannose, they are transported along the concentration gradient, as we can see here in this diagram, with the help of a glucose transporter GLUT5. The same transport can also be used by glucose and galactose if the concentration gradient is favorable. Glucose transporters. There are different types of glucose transporters that work along different cells and at different locations. Most important of them are 7 glucose transporters, and here are given 7 transporters and their corresponding cells and locations from where they work most of the times. Out of these 7 transporters, glucose GLUT2 and GLUT4, they are very important. GLUT2, especially, for example, they work in intestinal epithelial cells. They operate in intestinal cells and is a unique. GL, GLUT2 is a unipore system and not dependent on sodium ions. Glucose is held on GLUT2 by weak hydrogen bonds, and after fixing, glucose changes configuration and opens inner side, releasing glucose. As we have already seen in the last diagram that using GLUT2, glucose is transported towards the bloodstream, and it does not need sodium ions for its transport. Then GLUT4, they operate in the muscle and adipose tissue. GLUT4 is under the control of insulin. Insulin induces the intracellular GLUT4 molecule to move to the cell membrane and increases the uptake. While other GLUT molecules, they are not under the control of insulin, like GLUT4 molecule. GLUT1 is present in red blood cells and membrane, work most of the time in membrane. However, GLUT1 work in most of the cells of the body. It is also present in retina, colon and placenta. It helps in glucose uptake in most of these tissues, which is independent of insulin. Similarly, GLUT5, it works in small intestine, sperms, kidneys, like we have already seen in the last slide that GLUT5, it works to transport fructose and mannose. And then comes GLUT6 that works in lymphocytes, and GLUT7 that works in liver. Here is another transporter, GLUT4, which is a glucose transporter in the cell, and transport glucose molecule out of the across the plasma membrane and transport glucose molecule inside the cell. Maintaining blood glucose level using through pancreas and liver. Glucose levels in the blood, they are highly controlled, and as having either too much or too little glucose in the blood can have health consequences. So glucose regulates its level in the blood via a process which is called negative feedback mechanism. An everyday example of negative feedback in the feedback mechanism is in our kitchen. For example, if we talk about a thermostat, and if we set a temperature of the thermostat to cook a delicious homemade, for example, noodle at 375 Fahrenheit, the thermostat senses the temperature. For example, if we set the temperature of the oven, the thermostat of the oven, it senses the temperature and sends an electrical signal to turn the elements on and heat up the oven. When the temperature reaches 375 Fahrenheit, the thermostat of the oven, it senses the temperature and sends a signal to turn the element off. Similarly, so this is the example of negative feedback mechanism. Similarly, the body senses blood glucose level and maintains the glucose temperature in the target range. The glucose thermostat is located within the cells of the pancreas. So it is the pancreas who controls the blood glucose level. After eating a meal that contains carbohydrates, glucose level rises in the blood. Ultimately, insulin secreting cells in the pancreas sense the increase in blood glucose and release the hormone known as insulin into the blood. Insulin sends a signal to the body cells to move glucose from the blood by transporting it into different organ cells around the body and using it to make energy. In case of muscle tissue and the liver, the insulin sends the biological message to store glucose away as glycogen. Glucose is converted to glycogen in muscle and in liver. The presence of insulin in the blood signifies to the body and tells body that glucose is available for fuel. As glucose is transported into the cells around the body, it means that the blood glucose level now drops down and is decreased. The insulin has a opposing hormone which is called glucagon. Now the signal is the decreased level of glucose in the blood sends signal to pancreas. Pancreas release another opposing hormone known as glucagon, and glucagon communicates to the cells in the body to stop using all the glucose. More specifically, glucagon signals the liver to break down glycogen. It signals the liver to do an opposing function as compared to the insulin. That is, break down glycogen into and release the stored glucose into the blood so that glucose levels stay within the target range and all cells get the needed fuel to function properly. Now comes absorption in the large intestine. The leftover carbohydrates, almost all the carbohydrates except for dietary fibers and resistant starches, they are efficiently digested and absorbed into the body. But some of the remaining indigestible carbohydrates, they are known as leftover carbohydrates, which are broken down by enzymes released by bacteria in the large intestine. The products of bacterial digestion of these slow releasing carbohydrates, they are short chain fatty acids and some gases. The short chain fatty acids, they are either used by the bacteria to make energy and grow, are eliminated in the feces, or they are absorbed into cells of the colon, with a small amount being transported to the liver also. Colonic cells use the short chain fatty acids to transport some of their functions. And the liver can also metabolize the short chain fatty acids into cellular energy. The yield of energy from dietary fiber is about 2 kilocalories per gram for humans. But it highly depend upon the fiber type also. Then comes glycemic index. The term glycemic index or glycemic response of various foods have been measured and now ranked in comparison to a reference food. Usually a slice of white bread or just straight glucose to create a numeric value called the glycemic index. Foods that have a low glycemic index do not raise blood glucose level, neither as much nor as fast as foods that have a higher glycemic index. So foods with a higher glycemic index would raise blood sugar levels. A diet of low glycemic index food has been shown in epidemiological and clinical trial studies to increase weight loss and reduce the risk of obesity, while type 2 diabetes and cardiovascular diseases also, they are reduced. The risk of these diseases are reduced with the help of diets having low glycemic index. Absorption of fructose. Absorption of fructose is simple. It does not require energy and sodium ions. It is transported by facilitated diffusion mediated by a carrier. It is done inside the epithelial cell. We have already seen in the last slides in the diagram. Most of the fructose is converted to glucose. The later then enters the circulation. The fructose does not directly enters the circulation, but it is converted to glucose and then it enters the circulation, the blood circulation. And pentoses, they are absorbed by a process of simple diffusion. Medical conditions that affect how carbohydrates are digested. For example, there are some important medical conditions, the clinical conditions, such as lactose intolerance. What is lactose intolerance? When people do not have enough of the enzyme lactase. Lactase is an enzyme that digests lactase, so that digests lactose. So lactose is not when sufficiently broken down, resulting in a condition which is called lactose intolerance. The undigested lactose moves to the large intestine, where bacteria are able to digest it. The bacterial digestion then of lactose then produces gases, which leads to the symptoms of diarrhea, bloating and abdominal cramps. So lactose intolerance usually occurs in adults and also in children and is associated with the genetic generations and race. Sucrase deficiency is the inherited deficiency of sucrose and also leads towards diarrhea and abdominal cramps. Galactosemia. Galactosemia is a genetical disorder that affects how the body processes the simple sugar galactose, a sugar that is part of a larger sugar called lactose that's found also in milk. So it is also found in milk, cheese, and other dairy products. So galactosemia leads to having too much of this sugar in the blood, causing complications like liver damage, learning disabilities, or reproductive issues. Then comes fructose malabsorption. This condition has also been called dietary fructose intolerance, just like lactose intolerance. It affects how the body breaks down the sugar fructose from fruits and vegetables. The fructose is also present in honey, in agave, and the processed foods. The symptoms include nausea, diarrhea, and chronic fatigue. Then comes mucopolysaccharidosis. Hunter syndrome is a type of inherited disorder classified under mucopolysaccharidosis. The name of the syndrome is Hunter syndrome, HUNTER. Okay, it typically begins between the ages of 2 and 4 years old and is usually called by a missing enzyme that does not break down carbohydrates. Physical abilities, appearance, mental development, and organ function may all be impacted by this disorder. Pyruvate metabolism disorder. Pyruvate dehydrogenase deficiency is the type of inherited disorder classified under pyruvate metabolism disorders. It causes a buildup of lactic acid in the bloodstreams. Symptoms may begin as early as infancy. They include lethargy, poor feeding, rapid breathing, poor muscle tone, and abnormal eye movements. Now, there are factors that influence the rate of absorption in a gastrointestinal tract. First of all, mucous membrane. If mucous membrane is not healthy, absorption will decrease. And properly, macronutrients and monosaccharides especially will not be absorbed properly in the bloodstream. Thyroid hormones increases absorption of hexoses and act on intestinal mucosa. So in case of thyroid hormones, absorption will be affected. Actually, the hyper and hyposecretion of thyroid hormones affect absorption mainly through the intestinal walls. Then comes adrenal cortex. Absorption decreases in adrenocortical deficiency, mainly due to decreased concentration of sodium. And similarly, interior pituitary, it affects mainly through thyroid hormones, as interior pituitary produces thyroid hormones and they affect the absorption of minerals and macro and disaccharides. Insulin, rather, it has no effect on absorption of glucose. And vitamins absorption is decreased in B complex vitamin deficiency. For example, thiamine, pyridoxine, and pantothenic acid. And inherited deficiency of sucrase enzyme and lactase enzymes, they interfere with the corresponding disaccharide absorption. Here, our lecture related to digestion, absorption, and absorption of carbohydrates ends. Thank you.